The wide amount of variation we see in the human population is not only due to meiosis, but it is also due to the process of crossing over. Today we're going to talk about crossing over, or the occasional switching of genes from one homologous chromosome to another. So Morgan also discovered that specific genes are located on specific chromosomes, like eye color for Drosophila on the X chromosome. And remember that two or more genes that are on different chromosomes separate independently. But what about genes on the same chromosome? So Morgan concluded that the gene for wing shape and the gene for body color were located on the same chromosome, so they do not segregate independently. And linked genes that are located on the same chromosome tend to be transmitted together. But crossing over can occur during prophase 1 of meiosis, during the process of synapse. So a single chromosome can change as it passes from generation to generation. Gametes with chromosomes that recombine through crossing over have sections on their chromosomes that are maternal and other sections that are paternal. For example, one chromosome might have wild type body color and black body color, and then straight wings and curled wings. But instead, we might find some wild type curled and black straight due to crossing over. So we would expect all of them to be heterozygous, but we do find some with recessive traits due to crossing over during meiosis. So we have this image during meiosis 1, where the crossing over happens between non-sister chromatids. So if we said the red is the maternal chromosome and the green is the paternal chromosome, we can see that we ended up with 4 chromosomes, none of which matched the original parent due to crossing over. So when we look at the crossover percentage, we want to take the total number of recombinations divided by the total number of offspring. So let's say we had 18 total recombinations out of 300 offspring. Then we multiply this by 100 to get percent, and we would get a total of 6% of our chromosomes had recombinations. We can look at this percentage of recombinations and determine how far apart genes are on a chromosome. And so we take a gene marker, so recessive traits that are expressed on the recessive phenotype of an organism, and they're used to identify other genes on the same chromosome. So the white eyes, which are recessive, informs us of all the other genes that are located on the same chromosome. So crossing over will alter gene linkages along a chromosome. So if new segments of DNA are exchanged at marker sites, mapping is impossible. And geneticists will determine the position of the genes by their crossing over frequency. So if the crossover frequency is 1, the genes are much closer together because it's less likely that crossing over will happen right between them. However, if the crossover frequency is 12, the genes are much farther apart because it's more likely that crossing over at some point the gene split between those two genes. So the greater the frequency, the greater the map distance. The crossover frequency of 5% means 2 genes are 5 map units apart. And we just draw this on and we would put a 5 between them. So they're just 5 map units apart, say between chromosome A and chromosome B. A chromosome with a crossover frequency of 16 would be a total of 16 map units apart. And let's say this is chromosome C. So special stains have been created to identify the position of genes along a chromosome. So looking at the crossover frequency between gene A and B is 12%, B and C is 7%, and A and C is 5%. So we want to create a genetic map. So we can assume that A is in the middle, because A and B are 12 map units apart. So we have A, B, and they're 12 map units apart. A is not in the middle. So they're 12 map units apart. Now A is 5 units from gene C, which is then going to be 7 units from gene B. So it just takes a little bit of problem solving to determine the distance between those different genes. And again, you can do this through trial and error because you can draw them all out. It's always best to start with the 2 genes that have the highest crossover frequency and put them on either end of your chart and then go from there and try and fit the other genes in the middle. But again, the farther apart the genes are, the greater the frequency of the crossover is.